Hi, this is Jason from GeneCodes Corporation. We are excited to announce the release of our latest offering, CodeLinker. CodeLinker provides you with a rich set of tools for analyzing, exploring, and visualizing your microarray or RNA-seq data. With CodeLinker, you can choose from K-means, Jarvis-Patrick, or agglomerative hierarchical clustering, and then view your results using one of CodeLinker's clustering plots. The color matrix plot enables you to easily visualize the values in a dataset. The scatter plot is used for the pairwise comparison of two samples or two genes and allows you to visually determine genes that show induction or repression. The coordinate plot lets you look at the expression profile over all of your test samples or a sample's expression over the genes of interest. With the centroid plot, you can visualize the exemplar profile for each of the clusters arising from the algorithm you used. The cluster plot lets you drill down into clusters and view the individual member profiles. CodeLinker gives you two types of tree plots to visually highlight clustering relationships. The matrix tree plot is a combination of a tree plot and color matrix. The tree reflects the algorithm used such that closely related genes tend to appear beside each other. The two-way matrix tree plot is useful for visualizing the results of two clustering experiments, one based on the genes and one based on the samples, simultaneously. The self-organizing map is a clustering algorithm that is used to map multi-dimension data onto a two-dimensional surface. The goal is to uncover some of the underlying structure of the data by grouping similar data items together in nodes. There are three plots that complement the self-organizing map. The centroid plot is used to see the profiles of the values in a data set associated with a particular node. The cluster plot lets you drill down into the self-organizing map cluster to view individual member profiles. The matrix tree plot uses a color matrix of values, typically gene expression levels, in conjunction with a tree plot. Principal component analysis is a powerful technique used mostly in exploratory analysis of data and in predictive modeling. CodeLinker lets you visualize your analyses with 6 different plots. The scree plot is a simple line segment plot that shows the fraction of total variance in the data explained by each principal component. The score plot and 3D score plot project the data onto the principal components in 2 and 3 dimensions. There are 3 closely related plots that display the individual elements of the principal components. The term loadings refers to the extent to which the original variables influence the principal component. The color loadings matrix plot displays these loadings as a tiled grid of colored cells. The loadings line plot displays the loadings as a connected line graph. The loading scatter plot displays the loadings in a scatter plot of one selected principal component versus another selected principal component. With Sequencer, we brought you the ability to run your NGS alignments and differential gene expression analysis through an intuitive graphical user interface. With CodeLinker, we bring you the same ability to analyze your RNA-seq and microarray data in depth from your desktop. You can use CodeLinker to find new patterns of gene expression that are predictive of the disease you are studying. Your data contain hidden associations that CodeLinker can help you uncover. Sublinear association mining, or SLAM, searches your gene expression data for patterns of expression which occur together more than would be expected by chance. You can use these associations to train an artificial neural network. Once you've taught the artificial neural network with your training data, you can then analyze new test data. The results are displayed in the classification plot. Each sample in your test set is assigned a classification: predicted, true class, meaning something different from your original classification, or unknown, which will assist in confirming or refuting your hypothesis concerning the data. The Integrated Bayesian Inference System, or IBIS classifier, is a method that uses Bayesian probabilities to look at the patterns in your data. IBIS can identify nonlinear and combinatorial patterns of gene expression that characterize different toxicity responses, disease states, or treatment outcomes. It can then be used to build classifiers to identify these patterns in new samples. It can also be used as a search tool to identify single genes and small gene sets that show interesting expression patterns relative to the sample classification. You can perform a linear discriminant analysis search to evaluate the ability of each gene to separate the data into two or more classes. You can then display the results of the analysis for a selected gene. The background color gradient represents the classification that IBIS discovered, and the plot displays the gene expression in spots with colors representing the initial classification that you gave. You can easily see whether the gene or genes you have focused on fit the pattern you expected. For more complex data, where there is no linear relationship, CodeLinker provides you with the ability to perform two-dimensional linear discriminant analysis, or even quadratic or Gaussian discriminant analysis. Visit genecodes.com/CodeLinker today to learn more about CodeLinker.